Hi, my name is Shelby Porter, and I decided to do my video bonus assignment on the topic of CRISPR. CRISPR is actually an acronym. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. What's this mean? Let me tell you. Okay, so in 1987, these scientist guys were looking through bacteria's DNA sequence. They were going through and finding all this information out so they could gather it all and put it in a database to study. As they were going through, they found this one part of the DNA where it was the short little area of it. I don't know how short, but it was the short little area where it had the exact same sequence repeated multiple times. And within those repeats, it had little random spurts of information scattered throughout the repeats. Had a sequence, random, same sequence, random, same sequence. Kind of confused them. They didn't know what it was. They didn't know what it did. They just decided to write it down. Good thing they did. Fast forward to 1995, they were studying a different bacteria, and it had the exact same thing. Again, they didn't know what it was. They didn't know what it did. Fast forward to 2007, when they finally realized what those little random spots of information was. Turns out, it's a virus DNA. That's right, virus DNA within a bacteria. Who knew? This bacteria had somehow found a way, when a virus attacked it, to take or absorb or copy some of that virus's DNA and integrate it within its own DNA sequence, so that the next time that bacteria had cells repeat, DNA repeat, or was transcribed or anything, that little section of virus DNA that it had a copy of would go throughout the bacterial cell, and if it saw anything that matched the code, the exact same code, if it matched exactly, it would cut it out. Get rid of it, trash it, get rid of it. It's a defense mechanism, which is kind of awesome and cool to think about how this organism was so smart to not only withstand the attack that was thrust upon it by this virus, but it was able to take a part of this virus and integrate it within itself so that it could protect itself again if it ever encountered that virus. It kind of reminds me of how our bodies work with antibodies and defeating diseases, but that's another topic. So CRISPR. It was discovered originally in 1987. They didn't know what it was until 2007. Fast forward to 2012 when people are still doing research on CRISPR. Granted, it's limited research. It's not really exciting anybody yet until 2012 when they realized they had a breakthrough. What if you could use CRISPR technology in any organism? Any at all? Oh my gosh, what a mind opener this is. They could take CRISPR technology and make it universal to fit any organism. And essentially you can, which is kind of brilliant. Like, for example, if you don't like your eye color, say you've got brown eyes or something, you don't like the fact that you have brown eyes, you could use CRISPR technology to go into the DNA in your eyes and CRISPR out the gene that makes your eyes brown and replace it with like blue or green or purple or whatever you want. The same thing for your hair color. The same thing if your tree outside, you didn't like the fact that it was too short or too tall. You could CRISPR its genes and change it genetically. However, there are other uses to this technology that are less superficial, like getting rid of genes that cause cancer or Huntington's disease. Or in one research study that I just read about looking for ways to CRISPR out the genes that possibly cause type 1 and type 2 diabetes. This could change the world. This could change the medical field. Jump to 2015 when this guy has a breakthrough on CRISPR. He's going through and doing CRISPR research on mosquitoes that carry the gene that allow the parasite that causes malaria to live within them. He wants to CRISPR this gene so he can get rid of malaria, which is good. However, when that mosquito reproduces and have an offspring, there's only a 50-50 chance that offspring's going to receive the CRISPR gene, meaning eventually those mosquitoes' CRISPR genes are going to die out if they're not replaced into the environment every so often. Now, it's not physically possible to round up all the mosquitoes in the world and CRISPR their genes so that there's no longer any malaria. It just ain't going to work. So he figured out a way to develop this new technology that he called gene drive, which is where he CRISPRs out, cuts out a piece of DNA he doesn't like, and right next to it, he adds a CRISPR gene. That way, whatever offspring this organism produces, in this case the mosquito, this mosquito is going to pass on not only its original gene that's been CRISPRed, but a CRISPR itself, so that no matter what genetic code the offspring has, even from the other parent, if it gets the gene that carries malaria, that CRISPR will go in and cut it out anyway. Because the CRISPR was passed on, not just the gene, but the CRISPR. This is kind of outstanding, especially when you're talking about mosquitoes that reproduce rapidly. If you put this technology into a mosquito and released it into the natural environment, this could cause massive change to the mosquito population, meaning within a few generations alone, that gene that they carry that carries malaria would be wiped out for good. It'd be gone. No more spreading malaria through mosquitoes. Theoretically, this is a great idea. However, actually putting it into practice, a little harder than he thought. Turns out, the study goes, only 2 out of 3 attempts were successful, and that was in a lab with perfect conditions. There's still a lot more research to be done on the topic of CRISPR. I just find the whole thing entertaining and interesting to read about. Especially when it comes to the fact that this technology could have detrimental effects to the environment. Yeah, you got rid of malaria, but what happens if you CRISPRed the gene and it affects the genetic code somewhere else? What if a mutation happens and now the mosquito is no longer edible to its predators, or becomes toxic to its predators and it stops getting eaten? The population of mosquitoes could grow exponentially. They'd no longer be a specific point where their populations could end. This could be detrimental, which is why I personally don't think it's a good idea to be CRISPRing genes and then throwing it out into the natural environment. There's a lot of good that could come from this technology. I think it's really interesting, and I think the possibilities are amazing, especially when it comes to curing diseases. The superficial stuff I think should wait until you're more sure about it, but the possibility that this can help cure cancer, cure Huntington's, cure malaria, cure diabetes, I think it's worth looking into. Granted, a lot more research has to be done, but more and more funding is being thrown into this market every day. I recently listened to a podcast about CRISPR on RadioLab. It was really eye-opening. The fact that this technology could have so many good aspects, but also some bad. It's just the fact that it's unknown. I think this technology has the potential to do amazing things for our world, and I think it's very interesting, and hopefully you guys have too. I'm going to type out my sources and where I got all my information from, hopefully somewhere below this video or somewhere you guys can see it. If not, I'll probably just edit it in afterwards. But I hope I explained CRISPR well enough for you guys to understand, and I hope you got excited about it as I did, because I find this whole topic very interesting, especially the fact that it could change genetics as we know it. It could change animals as we know it. It could change our environment as we know it. Maybe for the good, maybe for the bad. That's up to you and scientists to ultimately decide. CRISPR. Clustered, regularly interspaced, short palindromic repeats. It's the future, and it's going to be interesting. Thank you, and I hope you learned something. Bye.